Hey everyone, welcome back to our channel Biomeds. In today's video, we are going to showing how to isolate a DNA from a bacterial culture. Let's get started. Before we are diving into the procedures, let's take a quick look at what you will need for this experiment. Materials: bacterial culture, lysis buffer, phenol chloroform, ethanol or isopropanol, centrifuge tubes, centrifuge and the pipettes. You will need a bacterial culture. This can be any bacterial strains you are working with. You will also need a pipettes for transferring the liquids. Bacterial culture is grown in an appropriate medium for 18 to 24 hours at 37 degrees Celsius. Let's get started with the bacterial culture. Once you have a bacterial culture, transfer a small volume into the microcentrifuge tubes. Use the sample around 10,000 rpm for 2 to 5 minutes. To the pellet, the bacterial cell discard the supernatant carefully. Now add a lysis buffer to the bacterial pellet and mix gently. Incubate the mixture at 37 degrees Celsius for 20 to 30 minutes. This is the critical step, is breaking open the bacterial cell and releasing the DNA. The lysis buffer, typically containing a detergent and other chemical to break down the cells, and after the incubation, add an equal volume of phenol chloroform solution to the lysate and mix it gently. Next, we will centrifuge the sample at 10,000 rpm for 10 to 15 minutes. After a centrifugation, three layers will form: top aqueous phase, middle layer and bottom organic phase. Carefully transfer the supernatant containing a DNA into a new clean tube. Make sure not to disturb the middle layer. We are going to add 2 to 3 volume of ice cold ethanol or isopropanol to the supernatant. This will help precipitate the DNA, making it visible as a stringy white precipitate. Incubate the mixture at 20 degrees Celsius for 30 minutes. Once the DNA has precipitated, centrifuge at 10,000 rpm for 10 to 15 minutes. Carefully pour off the alcohol. We will dry the pellet briefly and resuspend it in a TIE buffer or orange-free water to store the DNA. I hope this video helpful you understand the basic of the DNA isolation from the bacterial culture. If you found this video helpful, don't forget to like, share and subscribe to the channel for this scientific tutorials.